Today we were going to cover monomeric and oligomeric enzymes. This is a very small topic of your enzymology or biochemistry. It will hardly take your 5 to 6 minutes or max to max 8 minutes. So let's begin with this. So, as we know that your enzymes are made up of polypeptide chains, each polypeptide chain is called a subunit, right? So, the polypeptide chain is called as your subunit based on the number of your subunits. Your enzymes are classified as your monomeric, dimeric, trimeric, tetrameric, and oligomeric enzymes. So, these were all your enzymes. So basically among all these enzymes we specially goes towards your monomeric and oligomeric enzymes in detail. So in the basic introduction of your monomeric and oligomeric enzymes, they are classified on the number of polypeptide chains present in the enzyme, right? So enzymes are the biological catalyst that speeds up biochemical reactions without being consumed. So let's begin with your monomeric enzymes. So monomeric enzymes are those which consist of only a single polypeptide chain. So they cannot be dissociated into smaller units. Very few monomeric enzymes are known and all of these catalyze the hydrolytic reactions. Now, what is your hydrolytic reactions? Basically, it is a fundamental chemical process where water is used to break down the chemical bonds of any substance, right? After this, monomeric enzymes contain between 100 and 300 amino acid residues and molecular weight in the range of 13,000 to 35,000. After this, a number of monomeric enzymes are proteases. They catalyze the hydrolysis of peptide bonds in other proteins in order to prevent them doing generalized damage to all cellular proteins. They are often synthesized and active form, inactive form known as proenzyme or zymogens and activated as required. Zymogen, as we know, they are the inactive precursors of any enzyme. Basically, they are cleaved to form a active enzyme. They are activated by removal of their peptide section. One very common example we are having is proinsulin. Proinsulin is converted into insulin by removing its 33 amino acid peptide chains. And as we know that digestive enzymes are produced as zymogens and they are then activated when needed. So most of them are synthesized and stored in the pancreas and then secreted into the small intestine where they are activated by removal of a small peptide section. Because the digestive enzymes must be stored as zymogens, otherwise they would damage the pancreas. And if we talk about your proteases, then enzymes involved in degradation of proteins are called proteases, or we can say that they are the proteolytic enzymes. Basically, they play their role in your digestion, blood clotting, and your immune response. After this, we are having such enzymes include the serine proteases, so called because of the presence in the active site of an essential serine residue. Serine residue whose presence is essential for your enzymatic activity. Moving forward to your oligomeric enzymes, as we know that oligomeric proteins consist of two or more polypeptide chains which are usually linked to each other by non-covalent interactions and never by peptide bonds. So the component polypeptide chains are termed as subunits and they may be identical to or different from each other. If they are identical, they sometimes called protomers. They include dimeric, trimeric, and tetrameric proteins, and their molecular weight is usually in excess of 35,000. And majority of known enzymes are oligomeric, like we are having all of the enzymes involving glycolysis processes, either two or four subunits. Your oligomeric enzymes, they often show cooperative behavior while binding of substrate to one subunit that affects the activity of other subunits. Basically, they are generally more stable and allow better control of metabolic pathways, right? So with this we have completed your monomeric and oligomeric. Basically we have seen that monomeric enzymes, they contain single polypeptide chain, while oligomeric enzymes, they contain multiple subunits. In this we have seen proteases, which are the most important examples of monomeric enzymes and are often produced as inactive zymogens, right? But in oligomeric enzymes, they provide greater stability and regulation. So basically understanding enzyme structure helps explain how enzyme function in living organisms. So with this we have completed this topic. So with this, let's end this session here. I hope this session was very useful for you guys.